for the dihybrid inheritance, we are going to discuss two examples. We are going to discuss two examples. One of the examples we are going to take from the animals, and the second example we are going to take from the plants. Now, in dihybrid, we take two traits at one time and we study them. Now, in this case, we are taking the example of a cat, right? And we are discussing two traits of that particular cat. One trait is its fur color, and second trait is length of ears. Right? We are discussing two traits. One is fur color and second is length of ears. Now there are two fur colors. One is brown fur color and other is white fur color. Now this brown fur color, this is a dominant trait. And the white fur color, this is a recessive trait. Right? Then the long ears. Now this long ears and short ears. Now when I am saying long ears and short ears, actually I am talking about earlobes. Right? The pinna of the ear I am talking about for the cats. In this case, this long ears, this is a dominant trait, and short ears is a recessive trait. Now, if you guys see that this is what? This is the phenotype of both of these organisms.
So how do you get two breeds for cats?
Sorry?
How do you get two breeds for cats?
Again, what do you do that you let them reproduce together for many generations. Now, you will not be reproducing the kittens which are born from the same parents. Right? You will take a brown and long-eared cat and with another, you see, parental cats which have the brown and long ears, their kittens, you see, when they will grow up, you will mate them together, right? So eventually you will get what? Pure breeds, right? After 2 or 3 generations. But as their gestation period and their time to grow mature and reach to the age of puberty is longer, so this experiment will take a long time, right? In other way, to make it a short time, to perform this experiment in a short period of time, let's suppose if you have a brown long ears cat, you can confirm that whether it is a pure breed or not. How?
By breeding it with bias.
By test cross. Understand? So you can find out that the organism that you are going to use in the experiment, is it true breed or not. Got it? So you can find it out and then you can proceed. This one is both recessive traits, so you already know this is what? A true breed. For this one, you can find it out by test cross. So you can save your time. Otherwise, you have to go for, you see, producing the, you see, mating brown and long ears from different generations together, eventually to getting the pure breeds. Now, as this is a dominant trait, I will represent it with capital letters. So I am writing capital B for this. This is the also a dominant trait. I can use any letter, right? Let's use capital E, capital E. Got it? Now these both are recessive traits, so what I will be using? Small b, small e, small e for that, right? Now what are these? These are genotypes, right? Next, after the genotype, what do? We make what? Gametes. Now if you guys remember, yesterday we discussed, if I start this B joins with this E, it will become capital B, capital E. One gamete, this one joins with this one, it will be capital B, capital E. Another gamete, this one joins with this one, it will be capital B, capital E. Another gamete, this one joins with this one, it will be capital B, capital E. Another gamete. Right? So instead of writing all these, they are exactly the same. I am going to write it only once. I will write it only what? Capital B, capital E, circle. Same process is for this one, right? All the gametes, they will be made what? They will be same. And they will be what? Small b, small e. I am writing it like this. Alright? Now, when we cross this brown fur, long-eared cat with white fur, short-eared cat, this will be what we'll get, capital B, small b, capital E, small e. Now this one will be what? F1 generation. And this cat which we'll get, it will have brown fur and long ears. But this is not the true breed. This is heterozygous for both the traits. Now this is the F1 generation. So what do? We always cross F1 with F1. So here, we always cross F1 with F1. But before we move forward, we have to make the gametes for these, right? So we have the genotype capital B, small b, capital E, small e. Let's make the gametes for these. This capital B will join with this capital E, so it will become what? Capital B, capital E. One type of gamete will be like this. This capital B can join with this small e. Another type of gamete will be capital B, small e. Alright? Then this B has joined with both of them. Its probability is finished. Now this small b will join with this capital E, so it will become small b, capital E. And this small b will join with this small e to make small b, small e. So these four types of gametes we get, right? Now let's make the genetic diagram. Now when we make the Punnett square or the cross, we simply call it what as genetic diagram. Now for that purpose, first of all, what do we write? We write phenotype. In the phenotype, what do we have? We have brown fur, long ears. So both of them are brown fur and long ears. Brown fur and long ears. I just wrote brown and long. Now, the genotype of both of these organisms is what? Capital B, small b, capital E, small e, capital B, small b, capital E, small e, right? Next, what do? We write the gametes. Now, the gametes for both of them will be what? This. Exactly the same. So, one of the gametes will be capital B, capital E. Next, capital B, small e, then small b, capital E, and small b, small e. Same. Here capital B, capital E. Capital B, small e, small b, capital E, small b, and small e. Understood, everyone? Now next step, what we are going to do? We are going to make the Punnett square for this one. I am using the lesser space so that we can perform the second example as well on the same board. So we have 1, 2, 3, 4. 1, 2, 3. Now let's suppose here we have male, here we have female, right? Now here it will become capital B, capital E, capital B, small e, small b, capital E, small b, small e. Right? Then here we have again capital B, capital E, capital B, small e, small b, capital E, and then small b, small e. Right? Now what we are going to do, this one then will join with this one, it will become what? Capital B, capital B, capital E, capital E. What will be the phenotype? Brown, long ears. Now instead of writing complete, what I am going to write? B for brown and L for the long ears. Got it? But when you will be writing, you will write the complete. Right now I am trying to save some space. This one will join with this one, so it will become what? Capital B, capital E, small e. Again it will be what? Brown and long ears. This one joins with this one, it becomes capital B, small b. Capital E, again brown with long ears. Right? Here it joins with this one, so it becomes capital B, small b. Capital E, small e. What will be the phenotype here? Brown and long. Now, we are done with this one, right? So we come to this one. Capital B, then capital E, small e. Again, brown, fur and long ears. Now this one with this one, capital B, small e, small e. Very good. Now we have the new phenotype, brown fur but short ears. For the short ears I am writing S. Alright? Now this one joins with this one, so it becomes capital B, small b. Capital E, small e. Again we have brown fur and long ears. Now this one joins with this one, so it becomes capital B, small b. Small e, small e. Now what is the? Very good. Brown fur, short ears. Right? Next, we come to this one. Capital B, small b. Capital E, capital E. Again what do we have? Brown and long. Now this one joins with this one. Capital B, small b. Capital E, small e. Again what we will have? Brown and long. Right? Now over here what do we have? Small b, capital E, capital E. Now again we have a new phenotype. Very good. For white I am writing W and L. We have white fur now and long ears. Another new phenotype just appeared, right? Sorry?
Yes.
Now this one joined with this one, so it becomes small b, capital E, small e. Now what will be the phenotype over here? Mughis.
So it's the white and the long.
Very good. It will be white and long ears. Now when this one joins with this one, it will become capital B, small b. Capital E, small e. What it will be? Brown and long. Right? Now this one joined with this one, it becomes capital B, small e, small e. So it will be what?
Small b, capital B. Small b.
Yes. No. This capital B, small b. Small e. I wrote correct. Right? Capital B, small e. So what will be the phenotype here? Brown and short. Alright? Now this one with this one, it will be small b, capital E, small e. So what it will be? White, long. And last one, small b, small e, small e. What it will be? White, short. Right? Now we have 4 phenotypes. out of those 4 phenotypes, 2 are parental types and 2 are new phenotypes that we have received, right? Now we are going to write them down. First of all, what do we have? Brown, long ratio, brown, short ratio, white, long ratio, white, short. Now, first one and last one, these are what? These are parental types. While these 2 are what? These are the new phenotypes, right? Now, let's count the brown long ones. You have 1, 2, 3, 4, 5, 6, 7, 8 and 9. Right? 9 are brown and long. Next, let's count brown short. 1, 2 and 3. Now, let's count white long. 1, 2, 3. And only 1 white short is there. So what did we get? 9, 3, 3, 1 ratio. Now this, yesterday as well we discussed this point. This proves one thing, that both of these traits, their genes are not present on the same chromosomes. They are not present on the same chromosomes. That is why they are not affecting each other. That is why they are showing independent assortment. They are showing independent assortment, right? Because these both alleles, these both genes are present on different chromosomes. Now, if let me tell you one thing. If this is a chromosome, and on this chromosome, let's suppose over here there is a gene for brown color, and there is a gene for length of the ear. If these both genes are present on the same chromosome, you will never get a new phenotype. You will never get a new phenotype. You got these 2 new phenotypes, right? You will never get the new phenotype in this case. Why? Where the B will go, there the L will go. Where the B will go, there the L will go, because they are on the same ship, right? They are on the same motorbike riding together. So what will happen? That there will be no variation in this case. But as both of these alleles are present on separate chromosomes, B and L, it can independently go into any other gamete. It can go independently go into any other gamete. Understand? So they assort independently. So that is why, what do you get? You get more phenotypes, right? Inshallah, when we are going to talk about sex linkage and autosomal linkage, you will understand this point more. Now, yesterday, you see the period finished, but I was talking about Thomas Hunt Morgan. When he performed the experiments on fruit fly, at that time he came to know that there is a phenomenon called linkage. Because when he tried to produce some traits that he took 2 traits of of the fruit fly, he crossed the dominant with the recessive and he got the F1. Then he tried to get the F2 generation. He did not get the new phenotypes. He said that according to Mendel, there should be 4 phenotypes, but he was getting only what? 2. Same parental types. Then they said, where we are going wrong? Are we doing something wrong with the experiment? Or there is some other phenomenon? Then they came to know that when on the same chromosome the genes are present, that phenomenon is called linkage. We'll talk about it in detail. Till here, everyone understood this, right? Now, the next example, I'm just going to start it, but you guys will finish it. I'm going to start it, you will finish it. That example is from the plants. Now, here the example is of tomato plant. Now, for the example of the tomato plant, we are again taking two traits. One of the trait is stem color. One of the traits is stem color, and second trait is shape of leaf. Now, there are two colors of the stem. One is purple color and other is green color. Right? While there are two shapes of the leaf. One leaf is like this. One leaf is like this. The other leaf is like this. Now, this leaf, this is a serrated leaf because it has edges, right? The other leaf, this is called as potato leaf. It has smooth edges. It has smooth edges. Now, what is the word used in your book for this? Now, they have used the word for the serrated leaf, they have used the word cut leaf. And for this, they have used the potato leaf. Now, let me tell you one thing. That this purple trait, it is dominant. And this green is what? Recessive. This cut or serrated leaf, it is dominant. And this potato leaf, it is what? Recessive. Now you are taking two traits together, right? So one of the plants that you will take, that will have purple stem and cut leaf. The second plant will have the green stem and potato leaf. Understand, everyone? Now, for the purple, you can use any letter, right? Let's suppose that you are using capital A for the purple, right? And capital B for the cut leaf. Now, these both are recessive, so what you will do? Small a for the green and small b for what? Potato leaf, right? Now, you have the genotype, you have the phenotype. Now, next step, what do? You make the gametes. The gametes will made over here will be capital B. Gametes made over here will be small a, small b, right? When you cross them together, you will get capital A, capital B, small b. This one will be the F1 generation, right? And these all plants, they will have purple stem and cut leaves. Alright? Now this is F1. Now I want you all to start drawing the genetic diagram in which you cross F1 with F1. You make the cross, you make the gametes for these as well. Exactly. You have to draw the whole genetic diagram like this one. And you also make this ratio as well. Alright? Right now, start. Everyone.